Okay, hello everyone. Welcome here to our Nanolive Biolab slash photo booth. My name is Davide Merulla, and I'm extremely excited to be here today with you for several reasons, actually. First of all, because today we are going to speak about this, our 3D Cell Explorer, our revolutionary technology. Think of the future. Future microscopy is actually now. And second, because I am a microbiologist, a molecular microbiologist myself. I have a PhD in molecular microbiology, and I have spent many years, transfecting bacteria, inserting fluorescent genes, or doing any kind of staining that could allow me to follow up my experiment during my previous work. And after this long period of staining and marking that could take even months, I spent even longer time in some microscope rooms in the dark looking at my experiments. And I always had this fear in the back of my mind of, seeing not what my experiment was, but seeing the results of the heavy manipulation that I had my sample to undergo just to be able to see something. And the reason why I'm extremely exciting is because thanks to our technology today, this is a thing of the past. Okay, so today we are gonna spend about half an hour together, plus about 15 minutes depending on the amount of the question, and the webinar is gonna start soon. Just a couple of 3 details more. First, you can interact with us thanks to a Q&A panel that my colleague Lisa Pollaro has just enabled now. She is in the back office taking all your questions. So please start to write the question. Meanwhile, I'm explaining. She will make a list, and then we will discuss those questions at the end of the webinar. But please just write them as soon as you have it, because if you have them at the end, we don't have time to set up the list. Second thing, there is, yes, the Q&A button is there, exactly in this corner. And we will talk about resolution today. First time we will talk about resolution is the resolution of your screen. You have, on the other side, a button on your screen to set the resolution of the video. Please put it at 720. As today I will share with you my screen during the presentation, and you want to see it as big as possible. For the same reason, don't be afraid to go full screen. You will lose the Q&A, but it will be back when you will get back to the normal vision mode. Okay, I guess that we can start. And let's start from the end, as usual. So this is the final results that we want you to get. So a beautiful 3D reconstruction of a cell, okay, 3D tomography done in real time at the push of a button and in a completely non-invasive manner. Okay. Hello, here I am. Good. So during this introduction, we will tell you something about Nanolive's technology and where do we position during the history of microscopy. Show you some examples, just a few of them, of application of our technology. There's many more to come, and every time we interview early adopters, they come out with new beautiful ideas. We will tell you something about what is holographic tomography and how we did develop the technology that allows us to do that from the roots to the final machine that we offer now. We will tell you how to get to infinite live cell imaging. And at the end of this introduction, we will speak a little bit about refractive index analysis and quantitative digital staining. Okay. And after this, we will start the practical part where we collect some images. Okay. So, spoiler. Okay. This slide tells you something about the history of microscopy. So, history of microscopy started here in the 16th century with the discovery of cells. Then researchers understood that cells and life were deeply correlated. So what they started to do was to increase the technology applied to microscopy to unveil further and further the details of the cell, looking at better and better resolution. Now, the problem, if you look at this curve, is that the more you want to go down in resolution, the better you want to see the details inside your cell, the highest is the complexity of the techniques that you have applied. Needless to say that cells are transparent, so whenever you want to go beyond a certain type of resolution, you have to start marking cells. And this is true until the last Nobel Prize winning technology. It is not possible to observe cells at the level of details that we are interested in without heavily perturbing the state of the cell, adding some type of fluorescent marker, or no matter which kind of dye. Well, this is true until yesterday. Today at Nanolive, we oppose to this trend of more powerful but more complex, more costly, and more invasive. And we offer a device, the 3D Cell Explorer, that allows you for 4D cell tomography, meaning 3D across time, at a push of a button in a non-invasive manner and at a fraction of the price. So this is the machine, the 3D Cell Explorer, and this is the results that you can obtain. This is tomography, a cellular tomography that has been done in 2013. It's been published on this Nature Photonic article, and it shows you exactly what the technology can do. Okay, so this is what you get. You get the microscope and the software. The microscope allows you for really have results in a few seconds. You take your sample, you put it under the stage, you push a button, and you're going to have super comprehensive results just in seconds. Thanks, the results are so easy to understand thanks to our software. Steve, please note that the license of Steve is lifetime. So for less than 20,000 euros, so 19,900 euros, okay, not US dollar or whatsoever, you have the entire package and you have it for life. So I was talking about comprehensive result. Let me show you what do we mean for comprehensive results. So this is a movie of a lymphocyte that has been taken with our 3D Cell Explorer, lymphocyte that has been exposed to a chemotherapeutic drug. And what you see here is the slow death of this lymphocyte across time. What you can easily observe is the high level of details. And please note that the colors that you see on this video have been added after the video has been taken, thanks to a process that it's called digital staining. Okay, I will show and I will explain you very long in detail how this process works. But to bear in mind that we can change those colors every time we want, and we can add it really in 5 minutes, half an hour if you want to be extremely picky. Okay, now such a powerful technique clearly can be used for many applications, and here we have a list of possible applications which comprehend we can use it for proliferation, differentiation, apoptosis, virus cell interaction, bacteria cell interaction, cell-cell interaction, autophagy, trafficking, remodeling, and in general, all those cases where you cannot touch the genetics of your model organism, and you simply cannot add any kind of fluorescent stain, for instance. So it's an incredibly powerful technique. So we are entering in the part of the technology. I will tell you something about how we achieved a marker-free microscope with such resolution. How we can get to infinite live cell imaging. I will tell you about the compatibility of our microscope with basically everything you can imagine. Tell you how we managed to get such a high frame rate, meaning that we can get one image every 1.7 seconds. And for image, I mean a complete 3D stack of your sample. So we can make very beautiful movies with a very high frame rate. And at the end of this introduction part, I will tell you a little bit about the refractive index analysis, and then we will introduce you our software, Steve, and then we will move on to the practical part. Okay. So as you can see here, and I hinted you before, cells are transparent, so they offer very little contrast, and this makes the vision of the details inside the cell extremely difficult thanks to traditional microscopy. The thing, though, is that every transparent material has a physical natural marker, and this physical natural marker is the refractive index. So to give you an idea of what the refractive index is, the best is to look at this example here. So you have a glass which contains air on the top and water on the bottom. Now, the lights going through those different media interacts differently with the media because of the different refractive index of the light. The outcome that you can see here is that the straw looks like it's broken. Now, if you are capable to measure the refractive index of this region and this region, and actually also of the glass itself because it's a transparent media, okay, you can reconstruct the 3D structure of this glass with air and water inside. And this is precisely what we do with our microscope, but we do it at a cellular level. Okay, so how did we achieve this result and how the technique works? So to measure the refractive index, we produce holograms. Holograms are 2D maps of refractive index. How this happens? We have a laser beam, a very low power laser beam that is split into one beam goes through the chassis at the end of the machine, and it's called the reference beam. The second beam is shined through the sample and then rejoins the reference beam at the bottom of the machine. Now, what happens is that when the sample beam goes through the sample, since the laser is in phase, so the front of the wave has the same length, at the same speed, progress at the same speed. When it interacts with the sample, part of this beam will be retarded because of the interaction with the sample. And then when it will conjoin to the twin beam at the bottom, we will be able to understand how much it has been retarded thanks to the interference of those two beams. Interference that is recorded on the hologram. Okay, so we are capable to measure and record this difference in refractive index due to the interaction of the beam with the cell. Now, we did say that this is a 2D map. We want a 3D map. To achieve 3D reconstruction, we couple this with tomography. Tomography happens because we have a rotating arm at the top of our microscope. This rotating arm, as you can see from the animation, shines the laser beam 360 degrees around the sample. Meanwhile, a camera is taking a stack of pictures at a certain degree. This stack of pictures is collected by the camera and processed by the software to give you this 3D reconstruction. So, to sum up, combining holography and tomography, we've been able to produce a 3D microscope that is completely marker-free. Okay, so non-invasive at all. Now, talking about non-invasive, we did say we don't need any marker, which is extremely important. But on top of this, and it's also very important to say, the laser is so low power that you don't have bleaching nor phototoxicity. And thanks to the collaboration that we have with EBD, our partner, we've been able to develop a top stage incubator, a top-notch stage incubator that can be placed directly onto the stage of our microscope and allows you to control all those parameters such as temperature, pH through CO2, humidity, oxygen level, and so on and so forth. And this allows you to have hours or even days or even weeks long experiment. In our case, last time that we tried this top stage incubator, we ran out of CO2 before having problem with any type of the experiment. Okay? So all this, it's available now. Okay, now we are proposing you a device that is also extremely powerful because it's compatible with all the standard you want to use, and it's extremely intelligent. What do I mean for extremely intelligent? Our microscope is self-adjusting. So the optics themselves are able to understand if something is going on wrong with your sample and compensate with this for these differences. This means that you can use several different supports like plates or glass slides, and if you have any type of evaporation during your experiment, the microscope will compensate for the evaporation and will allow you to carry on until the end of the experiment with no problem or whatsoever. On top of that, we have ample space at the access to the stage, so you can really enter either with our top stage incubator or with whatever type of technology that you wish to apply. Okay, so one other very important aspect of the 3D Cell Explorer is that you can image within seconds, and this is possible because the structure of the microscope is extremely rigid. Extremely rigid means that we can really have high speeds to collect images. This result has been obtained thanks to a huge technological effort that led to the production of a unibody microscope. So we have one single piece of aluminum that goes through all the chassis that increases rigidity, lower size, lower weight, and really offers you an incredibly useful device. On top of that, we coupled this beautiful chassis with a very powerful GPU that is able to do all the calculation to transform the holograms in a proper 3D reconstruction in a matter of seconds. So altogether, we really can go down to 1.7 seconds per frame, which is really amazing. Okay, so when you shoot and you collect a 3D stack to make a 3D reconstruction, this is what you get, okay? This image is based on the refractive index. So you have a 96 Z stack of images with a resolution of 200 nanometers on the X, Y axis and of 500 nanometer on the Z axis. Since this is a Z stack, The only difference with the normal type of images is that normal images are based on intensity of light. We are based on intensity of refractive index, right? So the Z-stacks can be exported in the most common picture format like RAW or TIFF, and then can be processed downstream with all the type of programs that you're normally using, like Cell Profiler or ImageJ or so on, so forth. Okay, whatever you call it. Also, the very, very nice thing that we can do directly with our software is that we can do quantitative digital stain. So the refractive index is an intrinsic property of the matter. It doesn't change. It's not relative like fluorescence. So what we can do, we can select a specific region, like this one, for instance, of the image and assign a stain, a digital stain to this region. This allows us to segment and select only those parts to, in the end, being able to get this beautiful 3D reconstruction that allows really to go and see what happens inside the cell. You can tilt the cell, you can really look it from all the side views. So we are, so how this is possible? This is possible thanks to our software, Steve. Steve stands for Software for Tomographic Exploration of Living Cells. Okay, last two words have been squeezed in, but it makes sense. It's cool and it's easy to remember, and it works extremely, extremely well. So I guess that we are good for this first part. We can move on and go to the collection of a sample. So here we are. So this is our microscope. You can see how small it is compared to my hand. You have here the knob for the Z, X, and Y. Very simple. The screen has been cut so that you can see here a white light image that we will use for the focusing. And those are the three buttons that we will push to collect either, to stop the white light image, to collect either a 3D reconstruction or to launch a time-lapse. Today we will not launch a time-lapse, we will just take a single 3D reconstruction. So here you have, we have some HeLa cells. Those are very known, well-known standard. So just very simply, you take your cells, place them on your stage. You can see there, this green light is just some green light that we use to illuminate the sample so that we can see during the focusing. Voila. We are pretty much in focus. Yeah, we can search for a nice spot like this. Focusing is not really important that it's so precise, because anyways we are taking a Z stack, so the focus just need to be somewhere in between. And okay, so ready, steady, go. Please wait a second. Look at the arm. You will see how fast it is. Okay, now it's spinning. You don't see the laser because it's just too low power. And off we go. So here we have our 3D, our Z stack of pictures based on refractive index. So we can change camera. This part, the machine, we will not use it anymore. This part here is the top stage incubator that we developed with DVD. I will come back later to that at the end of the webinar, and then we will have a specific webinar upon it later on. Okay, so here we are. Steve meet people, people meet Steve. Steve, it's our software, as I told you before. It's extremely easy. There are three main panels. This panel here on the left is where you have your bright white light at the beginning for the focusing, and then you have your Z stack. You can scroll through the Z stack very simply. Let me increase the overlay so the image. So let's start from the top. So now we are at the top of the cells. You see almost nothing, and then the cells are slowly appearing, going down. We are at the cross section of the cell. You can already appreciate the detail. We can even zoom in, so we can see some vesicle, the nucleus, the Golgi, some membrane. And we can go down, 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 until going out of the cells. Okay, so let's go step by step. This other panel here is the panel that we will use, that we are going to use for the staining, the digital staining. Here you will see which regions of refractive index and gradient you are going to select. It's probably easier done than explained. So let's say that I'm interested in this vesicle here. The only thing I have to do is to go down here, select a color. We like to do vesicles in red. And what you see is when I'm moving the lens over this left panel, the central panel has some pixels lighting up, okay? This is because the lens is touching some pixels with a specific refractive index or index gradient. So what happens if I go here and I paint those pixels is that all the pixels in the area that have that specific combination of refractive index and gradient are highlighted. If you are not happy with how the selection was, you can move it afterwards. You can move it up and down. You can change the size. You can shorten it. Also, you can increase the opacity and the edge soften, but I'll show you in a second. So, thanks to the overlay button here, you can really follow in real time where your staining is applying. Okay. Now, the very interesting part is that this stain here, it will be reflected in the 3D segmentation. So let me increase the opacity and the softness to the maximum, so we will see those vesicles very well. And now, welcome to your 3D reconstruction first step 101. So we've been able to select all the vesicles into the cell and mark them in a selective manner. And I mean, this marking here took us how long? 30 seconds instead of months. Now, since we've been talking about marking, let me tell you that this panel here can be saved just like pushing this button, okay? Or even better, can be, after you record it, can be applied again to your sample over and over. So you can go here and select, this is a stain that we prepared very early this morning. You can just double click and boom, you have your stain applied in a fraction of a second. And this is what you can obtain just by a couple of clicks. Okay. Now, let's, before playing more with those images, let me tell you something more about this stain. This stain, the refractive index, is very different from fluorescence. Fluorescence is always expressed in arbitrary units. The refractive index is not arbitrary, okay? Once you have defined your stain, this stain will apply every time. So let's go and play a little bit with what's going on here. I can start from here. So I can, from the bottom here, we have the 5 stain that we have applied right now. We have the membrane that we can silence. We have the nuclear membrane that we can silence as well. We have the nucleus and the nucleoli. So you can see we're starting from the bottom. We can add details, increasing the amount of details that we want to see. We can play around with and go on and on. But the cool thing is that then we can twist ourselves, okay? And this is where the fun part begins. I could really play hours. You can, if you want to see it better, you can just push this button. You can really play hours and hours going through those cells. So, for instance, if you are interested in the membrane, you can really select your region for the membrane. We have this marking here, and you can really appreciate how moving through the cell. So I'm here at the top of the cell. You see the membrane is closed on the top, and then you go down. It moves slightly to the borders, goes down, 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 down. We are at the cross section of the cell. The membrane is all outside, and then we go down, 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 and we go through the cell, and the membrane closes at the bottom. This is just amazing. Now, there's many things you can do, and some are very easy and simple to do. So say that you want to just produce a quick pictures of this panel. You don't do anything else than go here, click this button, and save your picture. So let's say gray webinar 2, because we have whatever. And then you can actually take another picture, and this picture say you want to see the overlay. Again, you decrease the overlay and you shoot, and you say gray overlay 2, which is the problem with the French keyboard. Okay, now this is just a PNG, JPEG format, but of course you want to be able to export your data, so you don't do anything else than go here, activate this panel, and save 2-dimension, 3-dimension, and choose the type of format that you prefer. Okay, same thing, you can do it here with this panel. You can take pictures and shoot them out. Now, as you can see here, probably this is a sneak peek, we can also change the background. So you can go here and select any type of background. And this, probably violet is not the best color, black is cool. You can really produce quite stunning images, highlighting only the part that you're interested in. Let's say that I'm interested in the membrane. You can hide everything and just play with the beauties of the cell. Now, another thing that we find very, very interesting is that you can cut your plan. So you can go here, down here, select this cutting plan, hold shift, and enter into your image. Okay, so let's place ourselves here. And off, boom. What we obtain here is a cross section of your cell. You can really see different layers outside, nucleus, vesicle, nucleol, ER, and etc. So this is a really, really, really powerful instrument and very easy to use. And now, after being playing a little bit too much with this instrument, sorry guys, it's so exciting. I am gonna tell you to really rush and write down your last questions because we are running out of time, and I will leave you with a small video about the genesis of the microscope, after which I will be back and I will answer live your questions.
Nanolive's story began in 2007 during Jan's PhD in physics at EPFL in Switzerland. Together with his colleagues, he made a fundamental discovery in microscopy to see smaller than the wavelength of light. The results were simply stunning. Neurons, sperm cells, and even bacteria could no longer withhold their secrets. For the very first time, they could observe them with the same resolution as the latest Nobel Prize technology, and all of this with the dynamic beauty of unimpaired living cells. They published these unprecedented results in Nature Photonics in early 2013, with a huge impact in both scientific and popular science media. This was the dawn of Nanolive. Still, a lot of work had to be carried out. The original prototype from the lab needed to become a real product, small, intuitive, and affordable. Therefore, we challenged ourselves to reinvent a microscope from scratch. This process took 2 years, during which 2 prototypes were built, more compact and intuitive to use. Yet we knew that we had to reduce size and complexity to create a microscope unified by one simple concept. We created the first unibody microscope in the world, entirely derived from a single piece of aluminum. With this simple design, the 3D Cell Explorer became thinner, lighter, and more stable, a product characterized by the precision and perfection of details that only industrially crafted machinery can do. Dr. Grasberger was the first to test this advanced prototype at Harvard Medical School in December 2014. With this prototype now fully evolved, simpler, more robust, and easier to use, we finally felt ready. All across the Americas, and throughout Europe and Asia, the markets were speaking a clear language. By spring 2015, dozens of units were pre-sold around the world.
Okay, here we are back. I will give you another couple of minutes to end up with your question, and I will show you a little bit our incubator. So this is the camera a little bit. The incubator that we developed with EBD is composed by the 2 controller block. One is for the gas mixing, the other one is for the temperature of the top stage incubator, which is this box here. Very simple and robust. Okay, so the only thing that you have to do is to take this box, slide it into the microscope. It's magnetic, so once it's in place, it's very simple to use. You can open it, place your sample in there. I cannot go too much over the camera, and close it. Okay, so now, thanks to this incubator that we developed in collaboration with EBD, you can control. There is a sensor for humidity and temperature, so you can really control the humidity thanks to this column here. You can control the stream of CO2, so you can have gas, pH control, and you can also control the level of oxygen and nitrogen that you inject into this reaction, this incubation channel. A very nice feature of this incubator is that you can also very precisely control the temperature of the plate and the temperature of the lid. This means that you can put a very small gradient, say 37 degrees and 2 degrees more here, so that you avoid the formation of moisture on the lid, and you can really carry on with many hours, say days or even weeks of experiment. Okay, so let's move on with the questions that we have. Few of them that are pretty interesting. So we have a question about the staining. Can you share and record your staining with the others? The answer is it will be soon possible. So if we go on Steve, there is this button here at the bottom, which is going to be activated soon. And this is a cloud service that we're going to provide. So where people can really record and share those stains with each other. So once you have developed a nice stain for HeLa and you have certified somehow, you can really upload it and download it, and everybody can refer at this stain as a standard. Okay, talking again about the stain, we have about a question about how precise is the stain. The refractive index is, as I told you before, a physical property. It's an intensive property, so it doesn't change with the conditions. And if you can measure it precisely, and we can measure it very, very precisely, you can really refer to it. One thing you can do, for example, on this sample is to, if you know your media, like we know that this is taken in PBS, and in PBS the refractive index is 1.333, you can set this refractive index like this. You select the color, you select this, you paint your background so that you have a good representation of the background. Then you can go here, increase the background so you're sure you are going to get all you want. Okay. Now that we have this, we can go to the information here. You can click this button and you have here the panel when either you can name your stain, but in this case what we want to do is to set the stain as background and set the refractive index of the media. So 1.33, 1.333. Okay. Now you can say, okay, this has been hidden by itself. And whenever we are now going to measure any, so we can actually, with this tool, we can select any pixels of interest. And whenever we are going to select this pixel, first of all, we get the position, but also we get the specific refractive index. Okay. Then we have some other questions that popped out. Another question is, yes, what is the XYZ field of view and is it fixed? Okay, so the XYZ field of view is 80 micrometer X and Y and 30 micrometer Z. You can actually see it better here. Let me remove, sorry, the cutting. And voila. You have here your Z axis, so 0, plus 15, minus 15. And here you have your X and Y axis. Okay. There is an objective below the stage. So it is fixed and it controls the field of view that can be used. Okay, so we have this objective here at the bottom of the stage, which is the only objective that we have, and it's a 60x magnification. It was chosen like so because it's a good compromise between magnification and dimension of the field of views, and also offers a very good resolution. It's part of the instrument and cannot be changed. Okay. Then I think that we are getting close to the end of this webinar, and let's take the last question. is the microscope compatible with gridded dish? Okay, so yes, the microscope is absolutely compatible with gridded dish. We use gridded dish as standard also in our lab, and this is the standard procedure that we advise people to go through to certify the staining against a known and recognized technique like fluorescence. So what you can do is to have your fluorescently targeted cells and carry on take an image in the in fluorescence and with our microscope, and really prove that the digital staining that you are applying is staining the very same thing that the fluorescent is doing. Another thing that you can do is use antibodies, or for also certifying this staining is to use not fluorescently labeled antibodies, but all nanoparticles labeled antibodies, okay, that are directly visible in our microscope. So time is really running short. I'm actually 1 minute long, so let me wrap it up. Thanks for being here. Thanks to all the people in the back office that made this webinar possible. And as a little present for you that have been so nice to stay with us until the end, I am going to send you the data that we collected today, and you can download our demo version of Steve on our site. On our site, you can also buy directly our microscope. Please go and have a look. And with the demo version and the data that I'm going to send you, you will be really able to play and create your own stain and really see how precise and how neat is the image, also even beyond the quality of this video. So for the time being, it's everything. I'll hope you to see you to the next webinar, and also to all the list of events that you can also find on our site. We have an event page, so we will partake several conferences. So please check it out. We may be very close to you, and I'll see you next time. Bye-bye.